Right. Thank you very much. Can you hear me from the back? Okay, excellent. Well, thank you very much for the kind introduction. Yes, so I'm from Taiwan, very close to Hiroshima, as well as Hokkaido. The weather is much better here. So today I will talk about a knotty protein, and tomorrow in the tutorial, a longer one, I will tell you really topologically knotty proteins. We are structural biologists, so we look at things in the nanometer scale, not directly by eye, but not so abstract as quantum knots. So that's the photo of north mountainous area in Taiwan. So yes, there's snow sometimes, not often. But what I wanted to give you an idea today is that we try pretty hard to understand how protein glycosylation contribute to their functions. And I understand the audience here mostly are either chemists, material scientists, or mathematicians, theoretical physicists. So proteins are probably a bit distant to you, but it's important to realize that our cells, as well as virus surface, there's a lot of proteins, but the proteins are covered by a lot of sugars. So they really look like this under the microscope. So although probably you have seen, and I assume all of us have vaccines against SARS-CoV-2, and the antigen that we inject into our body, either mRNA or protein, they are the spike proteins of SARS-CoV-2. It is sort of sounds straightforward. We have the antigen, our immune system will have antibody, and they will see each other. But in reality, the virus are not as big as this, but our cells are really big in the nanometer scale. And how to find those targets that are highly glycosylated, so they're basically camouflaged by a layer of sugars, how do they find each other? It's still a very challenging task, but they actually do this very efficiently. So again, a big part of my lab is working on these topologically knotted proteins. They're literally knotted, and I'll show you how they knot tomorrow and why they knot. There is another, what we call, in the context of SKCM², knotty proteins, because they're really problematic. On the other hand, there is a big part of SKCM² is chiral, and proteins by default are all chiral. So in a way, we are working on chiral systems, no matter what type of protein we work with. So many of you know coronavirus, and this is actually the electron micrograph that we collected in our EM facility. It's not a human coronavirus, but it's very close. And you can see that there's a very well-defined double layer. That's the lipid bilayer. Outside, you see some fuzzy images. These are actually the spike proteins. And the reason why they are called coronavirus is because these spike proteins on the surface of the viral particles look very much like sun's corona. That's the Latin name. So that's why they are called coronavirus in the beginning when they are discovered, because these protrusions or these proteins really look like sun's corona. And that's the picture you will almost see every day in the past few years. That's the spike protein that we draw it artistically, but in fact, they look very fuzzy. And there are 4 structural proteins in the viral particle. The most obvious one on the surface is spike protein, but there are also 2 other membrane proteins called envelope protein and membrane protein, not very creative. And then the nucleocapsid protein, N protein, that's actually the target that we do the rapid flow. We poke our nose and try to see whether we're infected. Actually, they're detecting the N proteins because that's the most abundant in the virus, but they are not exposed to the surface. So if you have ever done this lateral test, you have to put some solution that's actually to break the virus apart. Then you can see what's inside, because it's easier to detect the M protein than the spike protein. Now, there are actually many different coronaviruses, and there are 7 coronaviruses that actually infect humans. What we don't want to see in the future is there will be some other coronaviruses that jump to infect humans again to cause pandemic. So those I highlighted in red. These are the human infecting coronaviruses, and there are ways to classify them. So we have the alpha coronavirus, beta coronavirus, gamma, and delta, etc. The ones we are really having issues with, SARS, MERS, they belong to the beta coronavirus, but the oldest or the first coronavirus that was identified to infect humans is actually the 229E that belongs to the alpha coronavirus. So this, you probably know, ACE2 is a protein in our body that's targeted by SARS-CoV-2. The virus somehow recognized the receptor ACE2 through the receptor binding domain here, colored in blue. And this binding triggers conformational change within the spike protein so that the virus can enter the cell to complete infection. So they are a large number of proteins encoded by the genome of SARS-CoV-2, but the virus particles themselves have only 4 proteins, as I just mentioned. But there are many, many other proteins encoded by the genome for various functions to facilitate replication, infection, etc. And because they're so important for structural biologists like us, we spend a lot of time to try to determine the structure as fast as we can, as complete as we can. So now there is a protein data bank. That's all the structural biologists, we deposit our finding there. There is a particular directory dedicated for SARS-CoV-2. So what you see here, actually, they come in different shapes. Some are on the surface, some are in the inside, some bind to RNA to replicate their genomes. But they are basically just proteins. There are many different ways to determine the structures of protein, and I don't have time to talk about this today. The ones you're probably familiar with, as at least I hope we learned this in high school or first year physics, diffraction. So Watson Crick in the 50s determined the DNA double helix structure using X-ray crystallography. And later on, there's NMR spectroscopy in the 90s to solve structure. And now the new kids on the block, that's a cryo-electron microscope. And I learned 2 weeks ago in Kyoto, Hokkaido actually has excellent setup here for the cryo-EM facility. These are the ones we have in Taiwan, Titan Krios, Talos Arctica, and they're really, really fantastic. What is very nice for us is that Academia Sinica is located in Taipei. Except the synchrotron, everything is available on our campus. The synchrotron is only 85 kilometers away, so it's less than an hour drive if we need to collect the data there. So it's really, really nice for structural biologists like me. And you are more than welcome to come to use our facility if you ever need such a facility, although Japan is also very, very well equipped. Now, you probably know we witnessed multiple waves of these variants of concern, starting from the Alpha variant, Delta variant, and now Omicron, and probably currently the dominant one is JN.1 strain. What do they mean in structural terms? That's because there are many mutations, primarily in the spike protein, that set them apart. So we have the Alpha variant all the way to Omicron, and these spheres indicate the positions of their mutations. Sometimes you see the mutation they draw as a single line and then multiple dots, but they actually fold into complicated structures. And you can see this is the receptor binding domain binding to the ACE2. In the beginning, very few mutations, but then over the time, as evolution starts, you can see that Omicron has many, many mutations within the RBD. And that's also why we need a second generation, third generation vaccine, because the prototype vaccine can no longer protect us because the virus mutate a lot. Now, I mentioned that most of the structural biologists or vaccine design principles are focused on proteins. And it is indeed very important to understand how the mutations change the protein structure and therefore the function of these viruses. So here is one example, and this is probably can be understood by chemists. So we have a particular mutation, asparagine to tyrosine. Now we have a phenol ring here. And then this introduces a favorable pi-pi stacking with the other tyrosine on the receptor. And a single mutation like this, we believe, is responsible for the enhanced infectivity of the alpha variant. So it does. So the spike protein has more than 1,500 amino acids. But a single mutation within RBD is sufficient to enhance the binding to the host receptor. And this is critical if we understood the molecular details of how these mutations change the structural function relationship. Now, in our institute or our academia Sinica, we have a lot of immunologists, antibody experts. So we collaborated with them. They have many monoclonal antibodies. So we solved the cryo-EM structures of antibody in complex with the spike protein, try to see why some antibodies are better than the others to protect us from getting infected. And there we can map the epitope, so the positions on the protein or the antigen that's recognized by a specific antibody. What we see here, for 3 different antibodies, they recognize different positions or different spots on the RBD. These 2 are actually quite far away. So we proposed, can we do an antibody cocktail? We mix 2 different antibodies that recognize 2 different regions, and they can bind to the RBD without clashing to each other, and basically covering most of the RBD surfaces to prevent ACE2 binding. This antibody cocktail also cleverly avoid this alpha variant mutation, so the structure that it recognized would not be affected because of this mutation. So that's what we did in the beginning of the pandemic, and that's very useful to provide the structural information to immunologists to develop better antibodies. The other thing we also did is we can use some biophysical analysis to monitor the binding between the spike protein and the receptor. And if the antibody binds to the spike protein and prevent the receptor binding, then we will see no kinetic response. But if a particular mutation that sort of destroy the binding affinity of the antibody to the spike protein, and in this case, the alpha variant of this particular mutation prevents the particular antibody from binding to it, and therefore you see that the biophysical measurements show clearly the binding events. We know the Omicron and other things, so we continue to work with our colleague on the campus to develop a broad spectrum antibody now can prevent different kinds of Omicron variants from binding to the spike protein, sorry, to ACE2. So these are all protein-based information. But in fact, as I mentioned in the very beginning, proteins on the cell surfaces and on the virus surfaces, they are highly glycosylated. What does that mean? They are modified by different kinds of sugar molecules. So you can see the gray or the salmon color, gray correspond to the sugar, but these sugars are not very rigid. So they're like the seaweed or kelp moving in the sea. So they are really covering a large space. So this is a molecular dynamics simulation. You will see multiple snapshots. Then these sugars cover a big part of the protein surface. So in a way, they covered most of the protein surface so that the immune system will have difficulty to see the protein surface. That's a way for the virus to evade from immune surveillance. In fact, it's not limited to SARS-CoV-2. So you can see that here we have green of different shades, and that's for the coronaviruses. The sugars accounts for 30 to 40% of the molecular mass of the protein. The record holder is HIV. Half of the molecular mass of the envelope protein comes from sugar. So if we need to understand how this protein works, we need to understand how these sugars work, or at least to see these sugars. The problem, or sorry, this is evidence that we can use light scattering methods to deconvolute the contribution of the sugar contribution to the total molecular mass. The thing is that for cryo-EM, for example, we try to get as high resolution as possible for the EM maps, but the sugars are very dynamic. So we actually have to do a data processing in such a way we preserve the low resolution information that you see in the transparent gray surface. And these low resolution information, we will see the additional blobs. They look like mushrooms. They actually correspond to the sugar structures that the computational people see in molecular dynamics simulation. On the far left, that's a snapshot of a single conformation. In the middle, that's a long-term molecular dynamics simulation. You take many trajectory snapshots, you put them together, you will see that they cover a big protein surface, and that corresponds very well to our low resolution cryo-EM map. So as I said, sugars are important, but for structural biologists, especially crystallographers, they need to be able to see the diffraction, meaning that within the lattice, every atoms are positioned in the same way. But that's inherently incompatible with the dynamics of sugars. So in crystallography, most of the time, people can only see the very bottom of the sugar, if at all. Even in cryo-EM, most people can only see the first two sugars of a long branch chain. So that's a problem. So here is an animation coming from UCSD. They use hundreds of GPU cards to simulate this dynamic event. You can see the protein structure shown in this ribbon-like structure. But on top of the protein, you can see all these cubes, spheres, triangles. They correspond to different sugar moieties. And they actually move a lot in solution. So this is what you see in silico. So you will see the protein start to move, but the sugars move even more. What they found in their simulation is that some of the sugars are responsible for pushing the RBD up. The RBD needs to go up in an open conformation in order to recognize the receptor. When it's closed, then it is unable to bind to the receptor. So this conformational transition is very important, and the sugar in this bottom area, they play an important role in facilitating the open conformation of these receptor binding domains. Actually, before the pandemic, we work on a cat coronavirus, and in our cryo-EM map, for some particular positions, we can see the sugar molecule very, very well. This is very unusual. But this means that if the sugar is part of the protein structure in a well-ordered state, you are able to see the sugar just as you are able to see the proteins. We can also see different types of sugar, but the resolution is not as good. So we will rely on mass spectrometry to tell us the molecular details or the chemical compositions of these individual sugars. We combine the chemical information we derive from mass spectrometry and the EM map, then we are able to build the atomic models of these heavily glycosylated proteins. But what we really want to see is how the sugars overall look like. So we need to do a lot of work to be able to robustly determine the chemical compositions of the individual glycosylation sites. What I show you here is a biological triplicate of 4 different SARS-CoV-2 strands, from green to yellow to purple to orange. It means different kinds of sugar. I will not go into the chemical details, but if you see, for example, this position is mostly green, it means that the chemical composition in that position is relatively homogeneous. If you have this it means that it's really heterogeneous. It comes in all kinds of chemical forms. And sometimes one experiment gives you this answer, the other experiment gives you that answer. It's sometimes not so easy to have statistical significance. So we develop a workflow to enable us to have really high statistical confidence to describe the chemical compositions. We can also see that for the wild type or the pseudo wild type and the alpha variants, the one particular position, the sugar compositions are very, very different. But in a cryo-EM map, they look very much the same. The protein look the same, but the sugars look very different. So how do we deal with it, or how do we describe this? So we teamed up with two collaborators. They were in Max Planck Institute of Biophysics. Now they are both independent group leaders in France and Poland. We have a software called GlycoShield. So what we do is we pre-simulate many different sugar chemical structures by molecular dynamics, but they are much smaller compared to the entire glycoprotein. So we can simulate microsecond trajectories for all of these sugars within a reasonable time. Then we have these ensembles we can then plant to the particular site that either experimentally we know the composition or we guess the chemical composition. Then we will be able to generate a fully glycosylated structural model. And remember the previous video I showed you, they have hundreds of GPU cards. Using this approach, you can do this in your laptop in minutes because we don't need to simulate the entire system. So it's like you're taking your favorite bonsai, put it on your balcony, and then you have multiple of these miniature plants, and then you can describe the thing rather than growing from scratch. With that, we are able to quantitatively describe which part of the protein surface is more shielded by the sugar, which part is more exposed for this structure. So here you can see if it's green, it means that they are more shielded by sugar, and in this particular domain here, it's not shielded at all. And it is kind of obvious that if a protein is not shielded and it is a foreign material, immune system will attack it. So indeed, we can have many different experimentally reported antibody-bound structure to show that this particular domain is more likely to be recognized by antibodies. So by using this quantitative description of how much the sugar shield your target protein surface, we are able to predict which part is more antigenic or which part is more likely to be targeted by the immune system. And that is very important for vaccine design, of course. So this is a overview of what we do in GlycoShield. Most of the structural biologists try their best to describe a naked protein. But these proteins are heavily covered by different kinds of sugars. And we are able to use this pre-simulated ensemble to plant them onto the protein surface using the experimental information we derive from mass spectrometry or other complementary techniques. Then we are able to generate a fully glycosylated structural models, and that will serve as a blueprint for us to better design vaccines or to predict which parts are used to recognize host. Because many of the viruses, it is unclear how they recognize the host. And it's also unclear which protein on the host surface is recognized by these viruses. So this computational tool, we believe, is very useful for many applications. Now, this is also online. So if you ever are interested in doing this, you can even use AlphaFold. You don't need to have your experimental structure. You can take any structure and try to see how they would look like when they have sugars. So glycosylation certainly is very important. And here is an example why. There are 22 different glycosylation sites on the spike protein of SARS-CoV-2. We saw many different variants, but the glycosylation sites are rarely mutated. it is very hard to remove the sugar and keep the spike protein intact. So they are important for functional and folding application needs. Only when it comes to JN.1, we now have 2 more sugars near the RBD. We are currently investigating the effects of these 2 new sugars. Maybe they are pushing the RBD further up to facilitate receptor binding. But here, you probably are not so familiar with that, but these spike proteins are not very easily expressed. The wild type or the original design, we get tiny amount. So protein engineers put different mutations. They are able to make them express better, sometimes 50 times better than the original one. But there is the stabilization of this very large machine has some serious implications. One is that having these 6 mutations change the glycosylation profile in this particular position, critical for the RBD in the upward conformation. And the result is that the mutant, when it turns green after this 6 protein mutation, so this is the original one, this is after the mutation, it turns from purple to green, it is less able to bind to the receptor, meaning that although you get a large amount of these spike proteins, but then the majority of these spike proteins, the RBDs, are closed. So it's a tricky balance. We need the protein to study its structure and function, but these mutants have different dynamics and different structure. So it's not so trivial to link directly to what they do. So there's still a lot of work we need to do. Here is another example that's really dramatic. The HKU1 is another human coronavirus, not so lethal, but it's probably most of us have been infected by this virus as well. Upon binding to a small sugar molecule here shown in pink, it would undergo a large conformational change. Here you see in orange. So it really opened up like this, but not directly in the binding site. Next to it, it opened up. What you see here, again, that's only the protein view. But if we simulate the glycol shield, you can see this protein has a very heavy coat of sugar. How does this conformational change takes place when there are so many sugars around? And in physical chemistry terms, you can imagine there's a lot of entropic penalties. You basically have to squash this very flexible sugar into sort of tiny conformational space and somehow change again. And this is really almost not explored. So one of our future research direction is to try to understand the physical chemical basis of this sugar contribution to protein conformational changes that are related to their functions. We can also do cryo-electron tomography. So you can see here is reconstructed tomograms of a virus. And then we are able, so this does not look very pretty, but we are able to reconstruct the shape of these viral particles, map out where the proteins are. Different colors means that they are in different functional states. So we can assign this by this relatively complicated data analysis workflow. The downside is that with Sinica, we can now work on human coronaviruses. So the biosafety does not allow us to do that. But here you can see that by using the best electron microscope we have, we are able to see a lot of details for the intact virus. Within the virus, we also see there is a particular sugar underneath this pink domain or the red domain. And if we change the sugar composition, so this is the peak coronavirus we identify in Pingdong, south of Taiwan. It's very dangerous for pigs, but not for humans. This is one from Colorado, where Ivan used to live. And this is another American strand. what we see here is that this Colorado strand has a mutation that removes the sugar. When we remove the sugar for our Taiwanese strand, then this domain becomes down. So there is a correlation, not a causal relationship. That here we can see the the D0 domain is down or up, but the population is higher for the up conformation for the Taiwanese strand. When we remove this sugar, then the down conformation, the population is increased. So there's a correlation that when the domain is up, these viruses are more dangerous. When the domain is down, the viruses are less virulent. We still don't know the exact receptor, so we cannot say for sure what it does, but you can see the sugar actually has an important role to maintain certain conformation, or actually large conformation of these protein structures. So this is just a quick summary. This is what I would say more than 90% of structural biologists are looking at, a naked protein. We can do better if we process our cryo-EM data in a different way. We can see the sugar, but the stem. So we now have a computational tool for us to visualize the fully glycosylated proteins in atomic detail. So with that, I just want to emphasize it's crucial to integrate different biophysical chemical tools with computational tools and working with medical people, biologists, to understand this better. And I'd like to thank all the people who contribute to this, especially SKCM², for me to be able to tell you this story. Thank you very much.